Hi, everybody. Thank you so much for being here. Today, we're happy to welcome Lhorane Lobjois from the Toulouse Institute for Infectious and Inflammatory Diseases, all the way from France. She has worked in the cell imaging core facility there for the last several years and is an expert in image analysis. So today, she's kindly agreed to give an introductory talk for QuPath and to demonstrate some of the typical image analysis workflows that she sees used. So thank you very much on behalf of the Early Career Working Group for being here, and I want to get started.
Hello, everyone. So thank you for the invitation, and I'm happy to share with you the QuPath pipeline. So I will share the screen. This one. Do you see it?
Yep.
Good. Yeah. So I will start with the QuPath and a nice video I put on this maquette. Just I would like to start by a quick reminder about QuPath if someone doesn't know QuPath. So it's a free software for viewing and analyzing 2D images. And at the beginning, it was more used for histopathology and wall slides, but now we use it also in fluorescence. I put different link, but I will come back on that after. And today, the last version was that I used is the 7.0, and you have the link here. QuPath is developed by Peter Van Kade, of course, and don't hesitate and don't forget to cite QuPath when you use it during your analysis. It's important. The QuPath user interface is something like that. So you can have the metadata of the image, the different annotation tools, the visualization of the object, the result, the scripting, also the link to imagery. And I will start with the brightness and contrast of the number 5. This one allows you to have this window, so brightness and contrast. You will see all the channels available. It's the same on Brightfield images. You can show hide. You can rename the channel and also change the color and play with the brightness and contrast. So adjust the histogram to have a good visual image. A reminder, of course, about detection against annotation. So an annotation, it's a region of interest, a flexible one. So you can move, you can change the size and do what you want with it. And most of the time it's drawn by yourself. And just there is a limitation about QuPath because it's a heavy object. So QuPath can handle like 1,000. It's quite huge, but yeah, it can be crush your QuPath. And the detection, it's a fixed object, lightweight. So QuPath can handle millions of them, and you can't move it, you can't change it. It's the end of your analysis. So most of the time, it's like the cell or the protein inside the cell. Also, there is class on QuPath. So a class is like a storage box for objects, so allowing you to label and organize them. You can create as many as you want, so that's great. And one object can have one class, but one class can have several objects. To classify it, it's quite easy. You draw an annotation, you click on the class, you set select, and the annotation will take the rules. To install more than QuPath can offer you, you have extension like plugging on imagery. So extension, manage the extension, you will have the QuPath catalog, so the extension already available on QuPath. So you can find a lot of them. If one is already installed, you will have this icon. If you want another one, you will have the plus. So if I would like to install something, I will click on the plus. I will have like this window. You can select the version to install and click on install, and it will be done just like that. And you can add more catalog, like the cell post catalog, so it's the BIOP. And thanks to that, you can have more and more extension like on VG. You have also help options. So for different, you have the QuPath tool. So on help, you have the QuPath tool, and it's like a short tutorial about QuPath interface. By different pages, you switch one by one and showing you a few tips and tricks also. You have the interactive help. So if you put your mouse on an icon, it will show you what it's do. So it's quite, if you forget where is the brightness and contrast, you can put your mouse and it will tell you. You also have the documentation. So you have the link on the beginning, but you have the access here on help with documentation. You will go on the website and you can find different tutorials, how to create a project, how to start with QuPath or do something more. Also, until the scripting and automation. So that's great. And if with that it's not sufficient, you have the image science forum. That's its community forum, I can say. Also, Peter Bankett is on. So you have the QuPath tag and you can find a lot of topics. I can't say how many, but you can add your topics and the community will help you. So it's a really good thing to have in mind. And I'm pretty sure, yes, it was the quick reminder and introduction. So today it's to talk about pipeline. So that's why I would like to show you this one. It's I tried to create like a mind mapping path about pipeline on QuPath depending on what I want and the question I have to answer my question, my biological question. So the beginning is to create projects. You will see everything after, but it's to help you to understand. So you will have the creation of the project, and if you want to create like a notation by yourself, the first question to ask is the signal is homogeneous and on all my images or no. So if yes, you will go there. If it's no, you will go there. And I will start by going on yes. So we'll go on this way. This icon represents the pixel classification, so we will work only on pixel, not on the object directly, and we will do a threshold. The goal will be to measure the surface of a DAB area inside a whole tissue, so I would like the surface and export my measurements. Yeah. So first things when you work on QuPath, it's to create a project. The project allows you to group images and process them in batch. It will save your annotation, your classification, the action you will perform on images. And the big advantage of QuPath is that you will store the path of your image and not the file. So you can duplicate the image 10 times if you want. It will not create a heavy file. To create the project, so there is one button, you click it. And then you can create or select an empty folder. It's really necessary to have an empty folder. If it's not, QuPath doesn't want to create a project. After that, you will add your images by clicking here. You can choose the file, and you will see the list of images you will import it on your QuPath projects. Then you can choose the image type. So it's bright field or fluorescence, and then you can import them. It's only that to create your first project. Then, as I told you, you can click on half moon, I can say. So the brightness and contrast. And for this first example, so I work on bright field and DAB. So I would like to show you the different things it's important to do when you work on bright field images like that. So you can open the bright field, see the different channel, and something like that. But at the beginning, it's not really separated. The hematosine can be on DAB and DAB can be on hematosine. So you will start by a color deconvolution. The color deconvolution allows the separation of color, as I said. So you will have a better segmentation and visualization of your signal. So it's important to do that. You can create a region of interest like that, and you take the background, it's necessary. And all your signal need to be in too. You will go on Analyze and Estimate Stain Vector tab. Yes, it's to say that the background is that, so it's good to say yes. And then you will have this window. So the goal is to do your stain vector length to be on the majority of the points. So to be faster, you have the auto button. You click on OK, and then you have this kind of results. So the segmentation, the separation of your channel will be better for the rest of the pipeline. So it's important to do that. And if I come back to the goal of this pipeline, it's to detect the DAB inside the whole tissue, so I need to detect my tissue. So for this, I will use a pixel classification with a threshold method. This is my tissue. If you don't know what is the threshold, so it's to to put a class for each pixel. So you will select a value. For example, this one is 100, and all the pixel above this value will be, for example, positive, and below will be negative, as you see here. So on QuPath, you have the classify pixel classification and create thresholder. This window will appear, and you have a lot of settings to do. So this is a result, but it's better to understand what I put here. So that's why I put it in bigger, but sorry, it's kind of not too sharp, but okay. So the resolution will be the level of detail of your objects. So if you would like to detect a tissue, you can stay on low or very low resolution. That means you will have a big pixel, virtual big pixel. It's not your acquisition. You can select the channel, so you can work on one channel or average channel. You can add different filter like Gaussian, Laplacian, depending on what you want, and it's available on the list, of course. This is the strength of your pre-filter add-in here. The threshold, so the value, and on which class the pixel will be. So on my side, I put below threshold the tissue and above ignore. I know it's not logical, but it's because we work on average channel. If we work on one channel, it will be the contrary. So sorry for that, but it's logical. And thanks to that, you can save it to apply it on all of your image and also create objects if you want. So just here I come back to show you what I have. So thanks to this, and the threshold quite high, with a big Gaussian filter, because I put a strength at 1.5, I can detect in black here the representation of my tissue. And because I don't want to have the hole, that's why I keep it like that. After that, I save it, I close the window so I can load it if I would like and apply it on another image, for example. So I am going on classify, pixel, and load pixel classifier. Then I can get my pixel classifier already created, and I apply it on all the image. Then I create objects, so I can create annotation or detection, but because you would like to detect DIB inside the tissue, it's necessary to create an annotation, because on detection, you can't have an object. It's the final thing. You can add filter like minimum object size. So if you want to remove small, some piece of tissue, you can. If you want to fill holes, it's also available. The rest, I will come back on that on another pipeline, so I will not showing you that now. And after clicking on OK, you see here, yes, I can play the video, the edge of my annotation. And thanks to the visualization annotation tool here, this is the edge, and the second one will be the fill annotation visually, so you can see what is on your annotation and when is your tissue, where is your tissue. So this is the first thing that I could do for the tissue detection. Then I need to detect the DIB. So this is a zoom on the image, so it's not so great, but this is a biological image. And I will use the same thing, the same method, so pixel classification with threshold. So I take a video to show you more dynamically, I can say, how to do. So you can go on classify pixel and create a thresholder. Yeah. You can adjust, by default, you keep in mind what you already do. So you need to change. And I don't know if you see, because it may be the lag, but on resolution, you have different kind of resolution. So now I'm going to work on cell signal. So it's quite, that you need details, so you can go on high resolution, but to not put your PC in fire, don't start by clicking on full because it's not great on each PC. Then I will choose the channel, so the DIB one, because I would like to work on DIB. So you see that the pixel work now, as you can. I can change the visualization to help me to detect the DIB. That's better than having whole. And I can adjust the brightness and contrast to help me to have the really DIB I want to see and segment it. Then I will change the classification because now I work on one channel. So below the threshold mean the white. So the things I don't want, so I ignore it. And the DIB will be above because it's a signal that I want. Then I adjust the threshold value. So on bright field images, the threshold is very, very low. So I mean small value like 0.1. And I adjust also the Gaussian to have smooth signal like that and not sharp. And my goal is to detect the DIB, only that. So I can adjust in real time like that. It could be long. And the things to keep in mind is don't doing it only on this area, but move on your sample to see if it's the same everywhere. And when you're okay, you can save it with a name you can recognize and not like test, because you will not use it well after. And then you can save it like that. You can close the file or going here directly to create the object if you don't want to close and load. You can choose where you want to create your annotation, your next objects. So you can work on full image, that means everywhere. And on all annotation, that means on annotation already existed your tissue. So you prefer for this kind of example, work on annotation and not have DIB outside. So you can say that. I don't know if I'm doing it. Yes. And then you can choose the type. So when I work only on surface, like tissue and DIB and have the big surface, I prefer to stay in annotation. But if I work on a number of cells, like I would like read the number of DIB cells, I will create detection. In this kind of example, I prefer annotation. Of course, I change the minimum object size because if I stay with 5,000, I will delete all my cells. So normally I put 0 or 5 to remove like 5 pixel dots or something like that. That is not a cell for me. You can split object at this time if you want to have each cell separately, but if it's only a big like that, you will have as one. So I prefer to not. Then I saved, I closed, and now you have your tissue annotation, you have your DIB annotation, and for each one, you can find the area just here below. And normally it's what I'm showing you. Yeah. So thanks to these two, you can do the ratio, for example, of the percentage of DIB on your tissue. To show the result and not click one annotation per annotation, you can go on the result table. So you have like this table here, and you have different measurement table. So here we are interested in annotation. So you click on that, and you can see one line per annotation. So you have your DIB and you have your tissue. If you don't remind which one is one, you have the classification. That's why it helps to classify everything. And you have the table is cropped, but you can find the area next on the next column. But it's only on QuPath. So if you want to export it as an Excel file and put it, I don't know where, but to do your statistics, you can export it, of course. But before each exportation, you need to save the project. It's necessary. If you don't do, the exportation will be empty. So you will go on measure, export measurement. You select the image you want to export the measurement. So this is not the example I take, but it's just to show you. You can choose the folder and the file name, which export you want. So if it's image, annotation, or detection, just to compare what is what. I just come back here. If you export image, it will be that. So quite nothing. If you export, yes, annotation, it will be there. So a big one. Yeah. The separator depends on you, if you prefer CSV or TSV. And you can choose the column of interest. So if you don't want to have an Excel file with, I don't know how many columns, you can only choose, for example, on this exercise, it should be image to have the name of the image, the classification to detect which one is DAB, which one is tissue, and the area that is below to have only that. If you want more, of course, you can select more. When you choose everything, you can export the result and it will be saved where you choose. So that was the first pipeline. So this. So I create my project. The signal is quite homogeneous. I create a pixel classification also for the tissue and the DIB. I take the surface of that so I can export the measurement. So I will go to a second one. And you see that it's quite different on this way because I had some lines and depend on your project. So it's an evolutive pipeline. So for the second pipeline, I would like to show you it counts the positive cell. So not a big one, but important, I guess. So first thing, it's also to create a project, but I will don't show you again. The signal will be homogeneous, I imagine, for this exercise. I will use deep learning and create a classification for the cell thanks to a threshold and export my measure. So for counting the cell, I need to segment the cell. So I will use instance seg extension. It's a deep learning tool for instance segmentation, so objects, in tissue and more dedicated to multiplex images. I will come back on multiplex after. This tool is developed by Thibaut Goldsborough and Pete Burnett. And to install it, so I already show you, but we can see it a second time, it's not a problem. So extension and manage. You have the instance seg with the plus. You select the version and click on install. And then you have it on extension and you can have as many as I said before. When you will run instance seg, you will have this window with a lot of settings. The first one is to choose the model you would like to use. At this time, there is 3, one for bright field, one for fluorescence, and one for single channel. You can run on all annotation on the image or only on the selected one or more. You can adjust these settings, so thread, tile size, and tile padding, it depends on your PC. So if you have a PC with a slow configuration, you can reduce that. If you have a big PC, you can improve that. The input channel, so of course, will be the one you want to work. So most of the time it's DAPI or DAB or something like that. So on these images that I will show you just after, it should be DAPI. And on the output, it will be single mask because we use the single channel. But if you choose another one, you can have the shape of cell or the nuclei, and you will already have the measurement of each cell. So I run it on this image, quite fast. So you can see the image with all my nuclei and my red cell. It's neuron culture. So here with the instance seg, you can see the segmentation of cell. In gray, it's easier to see. And instance seg, it's quite great and fast. So it works on each PC. It doesn't need a GPU. So it's really nice. And you see that small and big nuclei are segmented and individually. Then the goal is to say which one are red. So the classification of the red cell. And I will doing that by the mean intensity. So I'm going on classify object because now I have objects, and on QuPath, this object is a detection. So I'm going on object and create a single measurement classifier. I work on detection because it's detection that instance seg creates. I choose the channel of interest, so channel 2, the mean intensity, and I adjust my threshold as the same for the pixel. I choose on which class the object will be go. so positive or negative. And I have a live preview, so I can see in direct live, and I can save it to show you what I mean. So on my object, it's the results give something like that. So in red, you have the positive cell, and on quite blue or purple, I don't know which kind of color is that, but it's the negative one. And what is great, it's if you click on annotation, you have the summary of what is inside this rectangle, yellow rectangle. So you have the number of detection, the number of negative cell, positive, and the ratio of positive cell on all the annotation. So it's good to have a quick idea of the presentation of your cell and the number per region. So you can imagine to do that on a tissue. It's completely possible, it's the same, but it will maybe take more time for instance segmentation, but the classification, it's quite fast. And to help you to determine which threshold value you can put, when you have your segmentation, you can double click on a cell, and on the result here, you will see all the measurements per cell. So you can adjust, thanks to that, the threshold. And it was really fast because on counting positive cell, you can't have more things if it's only measure the positive cell for one channel. So that's why I would like to present you a bigger one, which is the most I can use on my lab. So this one is a different way to obtain your results. And it's a multiplex tissue. Yes, the time is great. I just checked. So for the multiplex tissue, you will again create a project. On this case, my signal will be not homogeneous, so I will need to create a training image. I will use pixel classification by threshold to detect my tissue. I will take also pixel classification by machine learning. I forget to add this arrow to get my germinal center. I will use instance seg to segment all my cells. I will classify them and then export my measurements. So I will have on a tissue the number of germinal center and the number of cells that will be, for example, positive for a signal on all of this region of interest. So I can start slowly. So the tissue detection by pixel classification with threshold, I already showed you before, so I can just put the video if it's not too fast maybe, but you can see how to do. So it's the same as in bright field. You choose average channel or all the channel dependent on your signal. You have a low resolution because it's tissue. The threshold on this one works well than on bright field, so you can have small value. And all above the value should be on tissue and below on ignore. Ignore, it's not a measure, so you don't have the value for the ignore things. So it takes less memory on your PC. And you can adjust like that the threshold in live. And then adjust the strength of your filters to add a smooth with the Gaussian like that. So it's less sharp. And then you can save it with a name. On my side, the pixel classifier, it's pxc underscore threshold, the number of the threshold, the name of the thing I do, and the version 1. Because it's quite simple on this example to have one version, but when you work on big projects, it's quite necessary to have a versioning, because one experience doesn't mean you will have a second one. So yeah, I prefer to add version. Then I create the object as I done before, and I can remove object if I want, but on this case, I don't need, and I don't have hole, so it's okay. And so now I have my tissue, yeah, and you see it by the edges here, and it's already here. Then I say that I create a training image because my signal is not homogeneous. So to do that, you have a classified training image option. And on this one, you have create annotation, region annotation. Because if you want to do a training image, it's like a patchwork. So you will represent all the heterogeneity of the experience. So if you have blurry, weak signal, absent signal, strong signal, you can have an heterogeneity. So it's better to have a training image for that. And you will not create your annotation by yourself one by one. That's why it's this option will do it quite automatically. So you can choose here what do you want for the size and where you want to classify them. So the width height, it's you that you want to use, but the size unit should be in pixel. By default, it's micrometer, but to avoid you to do a short calcul, prefer to stay in pixel. The classification will be in region. So also because I don't know if it's, yeah, I will say star. With the star, it's not included in the measurement. So that's why we use it for that. And you can create region as many as you want, but you need to keep the same number of this region per images. So if you have 10 images and you create 4 images on the first, you will need to create 4 small region per image. I say that because after you will create this patchwork of image, so the mix of all your little region. So on classify, you have training image and create training image. But before doing that, that's why I added that, because you need to save the project if you don't want to lose region. Then this window appears. You get the region thanks to classification. And the width of your image will be the width coming from the number of region you take on one image. So if I take, for example, 1000 for this one, I have 4, so the width will be 4000. If I take 2, it will be 2000. And thanks to that, I will have one line, one sample. So I clearly see all the heterogeneity coming from my experience. And then I click on OK. And on the list of my image present on the project, I will have a new one named sparse image with the number of regions you used. To show you the creation of a training image, you choose the type of the image here and inflorescence, and I apply, and this is the kind of image you can have. And you see the name here, sparse image with 8 regions. Of course, I will never take 2 per image in my analysis, so it's just an example to be faster. Then I would like to do the segmentation of the cell. So I will use again instance seg extension, but this time I will use the fluorescence model. I use fluorescence model on this one. Maybe the video can start or no, maybe by myself. Sorry for that. It's internet issue. Yeah, maybe it's really work. So instance seg work on annotation. So that's why yes, I can start and play. That's why on annotation, you have create full image annotation, and then we create an image for all the, an annotation, sorry, for all the image without drawing by yourself. So it's quite great, like that. I come back to extension instance egg. Yeah, I choose the fluorescence model. I adjust that. So for the input channel, you see all the channel because it's a multiplex one. And instance egg, I can't say header, but like to work on all the channel because it will help you. I just remove the channel that don't help me to recognize the cell. So that way I remove some of them. Output should be one or bus, nuclei cell or bus. So I prefer to choose bus. And then I run on the annotation already selected, and it will be processed for him 15 times, but it's a virtual decomposition of your image. And then you see the result. And it's not so bad because you see the signal, and instancing it's quite strong on multiplex. It will use all the channel to detect your cells. Then I will classify them by machine learning this time. So on classify object, you have another option than create a single measurement classifier. You have the train object classifier. For this one, you will have other settings. So the classifier, you have different choices. On my side, I prefer random trees. It gives me better result than the other one. you have features. On features, it's all the things you can add on your training to help the model to recognize your cell. So you can find which channel you will use, so one or more. You can choose the different filters, so not only the Gaussian, but you can use more, one or more. You can use one or more strengths for this filter, and you can have an idea of what's going on. And after that, you can do it on point only. That means you will just click to say yes, no, and see it in live. So I will show you how to do that. So on the features for this kind of measurement, I would like to say to extract, I can say, the pCNA positive cell. So I can enter the name, select all of the things linked to pCNA, and then I add all the shape measurements, so area, length, circularity for nucleus, cell, and everything. I create two different counting, so annotation tool. With one is set to pCNA positive class, and with one another, it's a pCNA negative class. So thanks to that and point only, I will just click like that to say all of them will be negative. And you see the counting increase. And it's depend of the user. So most of the time I'm doing that with the researcher or the doctorant on that. Then I switch to positive cell. The goal is to have minimum 7 or 6 points to start. Is the minimum to work. And when you have the same number, you can click on live and see it. So you see live what you prefer and what's done. So that's great. And you can adjust it. So if it's not great, so for example, some are for me negative, but maybe not, I adjust it and I see it in live. And you can do it as many as you want. It could take a lot of time, but it's necessary to do it the best as you can, because when you will apply that on all your images on your project, imagine you have 100 images, then you should be happy after when it works well at the beginning. And maybe the video is quite long because I do a lot of example with plus and negative. The goal of this circle is to have quite almost, if it's possible, the same number per person per class. So if the 50-50, it's not the representation of the number of cells on the image, it's the number of dots the model learn. So if you have the 50-50, it also recognizes at the same percentage the positive and the negative. So it's a good model if you have something like that. Then you can create more than one object classifier. So you can create one for pCNA, you can create one for small and big nuclei or other channel. So you can, thanks to that, after create a composite classifier. It means that you have one or more filter, most of the time it's for more, and you can combine them to create a composite that will use all your filter, all your things to detect the double positive, the triple positive cell. So for doing that, you're going on classify again and object classification and create a composite classifier. You select the different, not filter, but classifier you would like to use. That's why it's important to name it correctly. You create a new classifier with another name. You can save and apply. And then I can apply it. So when I apply it, I have something like that. The color are random. Sometimes it's the same, sometimes it's not. But if you see on the list here, I don't see the double positive. So KI is 67 and pCNA positive. So I need to get it back from the object on the image. So on this option row, you have populate from existing objects, all classes included subclasses. I would like to keep what is already here. And now I can see all the class newly created. So you can put a color as you prefer for you. For me, double positive is most of the time in white because it's visually here. And then you see on my training image if it's great or no. If it's not, I can come back to my classifier, create a version 2, and continue to train something like that. And on my annotation here, I have an idea of the number of the detection on this training image with the double positive, the PCNR positive, KI67, and the negative for both. So after that, on my big tissue, I would like to detect the germinal center by pixel classification with machine learning because I don't show you that again yet. So yes, I don't show you the path, but it's on classify, pixel classification, and you have pixel classification by machine learning or something like that. And you have a lot of settings again. So it's quite similar to the one we see. So the classifier, as I said, my preference, it's random tree. The resolution depends on the signal you want to detect. So it's a virtual resolution, as I said. The features, but if you don't know what the feature will done, you have the show button that you don't have for objects. So this option, when you select, will show you an image window with all the features applied on the image, one by one, and you can adjust the selection. The rest is by default, it's great. And also here you have the representation of the percentage for the training. So the goal, if you have 3 signals, is to have 33% for each. So here I choose already my features, my resolution, because the germinal center, I'm sorry if it's not that, but this round thing is the thing you want to detect. So moderate is quite okay. And the goal now is to draw. With the brush, for example, it's good. You can also use the magic wand. The both are great to do that. So the goal is to say, this one I want or I don't want. So I draw, I put on a class the annotation, I draw, I put in a class in the annotation to have what I want. So that, and I continue to do it, maybe 3 by 3, and sometime at the moment I will doing a live preview to know what I will have. Yeah. And then I see. Sometimes it could be long, and it depends on your resolution you choose and your PC, the capacity of your PC. This slider is the transparency of what you are doing, so annotation, for example. And you can adjust as the object classification, the result, to have what is better. And if I'm doing this same with the threshold, you can try it if you want. It's a imaging an image coming from demo website, but not the QuPath, another one. And if I'm doing it with the threshold, I can't separate each germinal center. So that's why I'm using the machine learning and to show you this option too. Then you can create objects, of course, because you would like to have the number of cells per object. So I'm loading the classifier, I create objects. I can work on full image or on annotation, but I already created my tissue, so I will stay on annotation. Then I create an annotation again, but for the brown objects, because I would like to have it and have Excel inside. I can, for this time, split object to have 1 by 1 objects. It's better to have a bigger one. That depends on you, but it could help. And then, after a few moments, I will see all the brown objects appears here. And you see that I don't remove a small object, so that's why I have something like it's not a good object. And you have option insert in hierarchy. That means if you don't create the object in a good way, so not in the order, I can say, you can click on that and you will understand which one is inside once, like the Matryoshka dolls, if you know what I mean. And you can see on each object annotation, you have how many cells inside, like a hierarchy inside. Of course, you can apply the classifier on the objects. It was already done maybe, but I would like to show you. So I load the composite classifier, and now I can see it on the whole object, on the whole tissue, sorry. And you see the double positive in white, the green, the magenta one, I guess it's PCNA if I remember well, and in dark red, the negative one. And you see the number for the whole tissue here. And of course, you have the area of the tissue, so you can do a lot of statistics after that. And yeah, no, it's not there. Okay, maybe after. You can visualize also the table of that. So you have the table results. So if you go on annotation, you will have the germinal center and the tissue, one per line. If you go on detection, you will have one line per cell. So sometimes the PC doesn't like to open it, but most of the time it's okay. Depends on your image, of course. Another visualization of the results is the density map, and I like it because it's clearly visual. It's different like that. So it works on detection. You can select the class you want to see. And if you remove just the visualization of detection, you have the representation of the, not the cluster, but where the cells are the more. You can change the visualization of the color. So like red, it's hot. So the more representation of the double positive cell. You have other density type representation, but honestly, I use only by area. And you can adjust the radius to see them and change the class to see where is your class. That gives you an idea and a visual idea of where the cells are. So yeah, I like it. And yes, rhythm table, I told them that. So on annotation measurement, as I said, one line per brown object with all the things inside. And all the things, yeah, all the measurements. You can add more. On analyze, you have add features and like intensity and shape, so you can add more. And on table result, you can, yeah, stop it. See, so as I said, the detection, so one cell per line with classification. You have the histogram representation if you click on show plot. And you can see an histogram with the mean of each things. So here it's the mean for the cell area in micrometer square. So you can change, of course, the measurement if you want to check. And also the scatter plot. It's from the 6th or 7th version, I don't remember, maybe the 6th. And on that, you can plot the distribution of your cell between two measurements. So that should help if you want to see something. And if I'm not doing wrong that, you can adjust, for example, a threshold classifier in live with the scatter plot that's showing you where the cells are. So that could help also. And yeah, this is the big one I show you. And the last is I would like to add this last thing quickly. Yes, I have 5 minutes again because we need question maybe. This is a scripting and automation thing. So after doing action on QuPath, you have on workflow tab the action you perform with the list of what you do. But not everything is there, so not count on that all the time. You can create the workflow or a script depending on what you want. Oh well, it's not great. Going on automate to see the script editor, you can find different scripts depending on project open and create short scripts like color deconvolution, create annotation from pixel classifier, and adjust the number. And thanks to that, you're going to run for a project, and you can batch for all the images. So that could help you because you're doing the script one time, and then it's finished. Yeah, I finished. So if you have questions, don't hesitate. I hope it was clear and not too fast.
Hi, Lorraine. Helen, did you want to talk about the questions? I can do it in case.
I was just gonna say thank you so much. And
No
yeah, we
problem.
do have some questions in the chat. So I think one of the ones I saw was that we could just start with is kind of deciding whether or not your image is homogeneous versus not. How do you kind of go about, looking at the signal and deciding what you're kind of looking for in that sense?
I can just stop the sharing. It's okay. Yeah.
Yeah,
Because I have a lag of internet, so I don't hear you well with the sharing. It's too strong for you. Sorry.
maybe just
I can.
yeah, for the first question, just kind of discussing like how to decide if your image is homogeneous or not and what kind of signals you're looking at.
Most of time to have an idea of my signal, I'll open one of condition, like 2 image per condition. So for example, I have a normal and a sick condition. I will open 2 by 2. And if I have like 2 experiments, I will have 2, 2, and 2 by 2. Like to have an idea of in control and in sick or in treatment or depend on your sets on your experiment, what kind of signal I have. And most of the time I perform my images or so I have an idea like that. But it takes time to have an idea and select your images. But yeah, I will say open few images per condition to have an idea. If it's the same, good. If it's not the same, you know what to do. You're on mute, yeah.
And there's another question here in the chat that says, which external tools do you recommend to use for annotation in cell biology if you're looking at membrane or nuclei?
On annotation tool for membrane, it's no, not on manual, but on AA tool for membrane signal, I will use cell pose extension. So for this one, it works well. I don't know if I can find an image, but I can send after if you want some links for that. You will need for that a big PC. Yeah. It's the one that work well. You can try instance egg for membrane. So on output, you will only choose cell, but it depends of the pixel size. For instance egg, it's 0.5 micrometer. If it's another thing that couldn't work. That's why I say cell pose for that. That could help.
Any other questions? Feel free to unmute if you want to ask yourself.
Yeah, someone had a question. Does QuPath handle overlap cells for segmentation, and how do you suggest to do that?
I'm just sorry for the beginning. I don't understand what you talk about.
Yeah, their question was, can you segment overlapping cells, and how do you suggest to do that? My first thought is watershed, but do you have any other suggestions?
Yeah, again, I will go to IA tool, sorry. But for overlapping, you can try StarDist. That could quite well for that. But StarDist, it's only for round shape, like nuclei. If you want to have a membrane, as I said, maybe cell pose or instance egg. But for round or nuclei, I could say, yeah, StarDist, and if it's not, try the other one. They are quite great IA tools. I'm trying to see if I have an example of that on the presentation to show you, but maybe not here. Maybe here to show you the cell pose membrane tool. Okay, can I share quickly? Yeah, this. So here it's an example on adipocytes. I don't know if it, yeah. And you see the cell pose, detect the membrane, and each lipid droplet are individual. Or here, it's from the membrane of the cell, so you see it's outside the nuclei, of course. So maybe that could help to answer the question and to give you an idea of what it means. And no, it's imaging. In this one, no. Yeah, because TARDIS is showing it on another thing. Yeah. But TARDIS is for nuclei, and cell pose should be both, but more used for cytoplasm or membrane. Instance seg, it's more dedicated to multiplex images, or if you have a slow PC, I can say, because it works for nuclei and cells, so sometimes it's the best way to start.
And you find that you typically use the QuPath extensions rather than, like, you find those are the most beneficial for segmenting what you're looking at?
Yeah, clearly.
Yeah.
There is a cell detection on QuPath directly integrated, but no, it's not so great, sorry for QuPath, but I prefer AI tool and extension for that.
All right, any other questions? Or otherwise, thank you very much. We're at time, so we appreciate you being here, and
Yeah,
thank you so much.
thank you for your attention. Thank you for your question and the invitation, of course. And wish you a good day.